Welcome you all to this Cryo-EM course. Today we are going to discuss little bit more about the graphene or graphene oxide coated carbon coated grid. Previously we discussed about the sample preparation using the high pressure freezing instrument or self pressurized rapid freezing techniques or freeze fractured FIB-SEM and discuss very little about the correlative light and electron microscopy techniques, but those techniques are getting very popular nowadays. Today's we are going to discuss little bit more about the graphene and graphene oxide coated carbon grid. So what is graphene oxide? So let's take a look. So what is the graphene oxide? So here I am taking these things from a journal where you can see how we can synthesize the graphene or graphene oxide. Generally this graphene getting a single layer graphene oxide grid, it is the most important part, because if graphene is folded you can see different types of folding of the graphene. One thing you just remember that graphene or graphene oxide, it is basically a carbon layer. So if you look at this is a single layer graphene where you can see carbon is sp2 hybridized and it is a planar, and it has a hexagonal shape and all having a carbon, and this carbon is sp2 hybridized, that is the reason it is planar, as well as it is very transparent, and graphene basically graphene or graphene oxide basically very stable. Now most of the time this graphene oxide it is forming this multi-layer graphene or graphene forming a multi-layer graphene. This multi-layer graphene, from this multi-layer graphene we have to isolate single layer graphene. If we get the single layer graphene, those things are very useful for different type of TEM and Cryo-EM study that we are going to discuss now. So when we have a graphene we can use, or when you have a graphene oxide, means graphene is the oxygen molecule is there. So this graphene oxide or reduced graphene oxide, how we can prepare. So sometime this graphene, that this is a sp2 hybridized carbon, right. Sometime this sp2 hybridized carbon will interact with the oxygen molecule and either it will form a epoxy kind of group or OH group or COOH group. So that is called the graphene oxide. So after graphene oxide, if you use different kinds of chemicals or heat energy, we can reduce some of this oxygen molecule, we will remove some of this oxygen molecule and we can form that reduced graphene oxide layer. So how we can prepare this graphene oxide sample? So generally if we have a graphene powder, so that we can use that Hammers method or improved Hammers method or modified Hammers method, where we can use that KMnO4 with sulfuric acid and the nitric acid, or the KMnO4 with sulfuric acid or phosphoric acid. So here you can use that KMnO4 with sulfuric acid or sodium nitrate. So in all the cases here we are using the sodium nitrate with sulfuric acid and 3 KMnO4. Here we are using the modified one, 6 KMnO4 with sulfuric acid and 0.5 sodium nitrate. Now using this we can prepare those kind of graphene oxide, that this method we can make it a hydrophobic carbon material that we can dissolve in solution and prepare different kind of graphene oxide sample. Here we are showing you that what is the structure of this graphene oxide. As I mentioned that it is sp2 hybridized carbon layer. So here you have a hexagonal carbon layer which is almost looking like a benzene ring, and then some of this group are oxidized because we modified with the chemicals, and after oxidation we are getting this kind of compound where we have that COOH group or epoxy group or So, epoxy group will be connected with this carbon layer where there is a 2 bond will be connected with the carbon with the oxygen, and the COOH group or OH group. Okay. In this particular class, our interest is not to how to synthesize graphene oxide or graphene, because there are lots of chemical processes that we can utilize. We can modify those graphene oxide with some other functional group, and that will really help us to prepare that modified graphene oxide or graphene grid. So, that is not our intention how to modify those graphene oxide grid. Here we are more interested how to prepare those graphene or graphene oxide grid, and how it will help us to coat our sample on top of that grid. That was our main interest. So, as I mentioned earlier that here our main interest is that how to isolate the graphene multilayers to monolayer. So, one of this study, so we are many groups are trying these methods to isolate graphene monolayer, because graphene monolayers, if it is a graphene monolayer, it will be much easier to coat the quantifoil grid or any carbon grid, as well as if there is a graphene monolayer, you cannot see the graphene at all or graphene oxide at all. You can, electron beam will be transparent, and graphene oxide or graphene is a transparent, and electron beam can go through that graphene easily. So, it is not going to affect our imaging, and there will be no artifact it is going to come, and as well as the graphene oxide is or graphene is strong enough, it can handle the electron beam. So, that is one of our main target, that we are not focusing on how to synthesize the graphene oxide or reduced graphene oxide or only graphene. Here our main target is to isolate that graphene monolayer or graphene oxide monolayer, coat our grid and do the imaging, and whenever we are doing the imaging, what kind of things we are going to focus. So, here you can see this is a graphene oxide solution, which is a 1% graphene oxide solution. So, we will take a 1% graphene oxide solution, that will be total volume maybe 200 microliter or 300 microliter. Then we use that ultra sonic water bath for the sonication for 30 minutes, because those kind of thing, that whenever we are using that graphene oxide solution from the sigma we can purchase, it will be always better to use the fresh solution, otherwise graphene oxide will come together, it will aggregate and it will precipitate, and that will be very difficult to use that water bath sonicator to separate them. So, why we are doing this water bath sonicator? Because we are trying to separate that thin film of this graphene oxide or graphene from the aggregation. Then when you have this solution, after sonication, will do the centrifugation for 10 minutes and precipitate most of the graphene oxide aggregates, and will take the supernatant. When we will do that centrifugation step for 10,000 rpm for 10 minutes, you can see there is a small pellet is deposited. This small pellet is deposited means it is size is very big, very large, and everything is precipitated. And the solution you can see it is not very clean, it is some kind of orange color it is visible. What we are doing, we are taking this supernatant and keeping it in the other side. So, large geoflake, whatever it is precipitated, we resuspended that flake with again it is a mixture of water and ethanol. So, we notice that if we just use the water molecule only, so the separation will be not that good. If we just use mixture of ethanol and water, the separation will be much better. So, here we are doing that separation mixture of this water and ethanol, and then we make that resuspended, and we repeated these things couple of round, and at the end you can see a very light orange color solution, where you can see that there is a separation of different types of graphene monolayer. when we have this separation of this graphene monolayer that we can use to coat the grid, we can use a 3 microliter solution and add to on top of the grid, and excess buffer solution we can blot it. So, here that I was discussing that when we have a geo solution, so when we have the only the water molecule, there is a high chance this graphene oxide there is a functional group, that epoxy group is there, OH group is there, COOH group is there, which will interact with the water molecule and it will not easily separate them. Where we have the OH functional group or COOH functional group, if we use the ethanol or methanol, we have a CH3 functional group which cannot form a hydrogen bond with the other functional group. That will basically help us to separate those layer properly. So, here we perform some kind of optical microscopy imaging also, where we can see the large graphene oxide monolayer, where you can see the size is almost 30 micrometer size, and 130 micrometer size is good enough to cover a good area. So, we can see lots of large flake and it is completely separated. So, when we have this kind of separation, we can use those kind of graphene oxide monolayer to coat the coat our quantifoil grid. Mostly we are interested about the quantifoil grid, because this quantifoil grid, if we coat with this graphene oxide layer, there will be a thin layer and that is a transparent. We cannot see that thin layer at all, and if we add the sample, protein or any biological sample will absorb on top of that grid. So, even there is a sample concentration is low, it will be helpful that protein will absorb more. So, we can even go for a very less amount sample concentration to image using the graphene oxide grid. Now, the other part is that when you have a graphene oxide grid, that okay, graphene coating is there on top of the hole. So, that will basically help us. So, we discuss about the beam induced motion, because our sample is staying inside the amorphous ice. When electron beam is going through that sample, there is a motion on that sample, and that is called the beam induced motion. That beam induced motion basically it is affecting our images, and it is basically affecting our high resolution signal. If you just focused on that, if there is a continuous drift, overall shape of the molecule you can visualize, but all the fine details it will go. But if there is a platform, so graphene oxide or graphene platform is there and sample is added there and electron beam is passing through that sample, that beam induced motion it will be not there anymore. That will basically help us to visualize the sample in a very stable condition. So, if you look at this particular grid, you can see here there are couple of different color circles. So, this is one of the square where you can see different circular grids, and if you look at this red and the yellow and the blue, you can see different types of intensity of that hole. So, if you look at this red hole and this nearby blue hole, you can see the intensity is, this hole is much brighter. So, that means this hole is completely empty. If you look at this yellow hole, you can see this is the partially covered with the graphene oxide. But if you look at all this blue hole, most of the blue holes is covered with a very uniform size the graphene oxide. This is basically some kind of graphene aggregation or graphene oxide aggregation precipitated here. But you can see majority of the holes are very stable, and then if the holes are stable, if ice is not melt easily, per hole you can use multiple images, per hole you can collect multiple images. So, here I am showing you one enlarged view of one of this particular hole, where we are just targeting one of the center part, and now when we collected this data, you can see this is a very less amount sample we are using, but the overall sample distribution is very nice all over the grid. Okay. So, now if you just evaluate that, different hole. If you just go here the previous one where I just mentioned that there was the blue holes are basically graphene oxide distributed nicely, and in this blue hole what we are targeting hole number 1, hole number 2 and hole number 3, and one of the yellow hole we want to image, that is hole number 4. So if you come here you can see this is hole number 1, 2 and 3. If you look at carefully, this is a low magnification. Here you can see this is basically that entire hole is not coated properly. So it is looking some part of this hole is folded. So maybe that graphene oxide is folded here. Here also similar kind of things happen, that graphene oxide is folded, or there is a part of the graphene oxide is broken and graphene folded in such a way. Now in this small magnification we cannot see it. What about if you go to a very high mag and if you look at this different hole, that okay, do we see a uniform distribution of monolayer of the graphene oxide, or do we see the multi-layers of the graphene oxide? That is the reason we targeted hole number 1, 2 and 3 and then hole number 4. So if you look at this hole you can see it that there are, I cannot see any multiple layers of the graphene oxide, but if you look at hole 4 you can see there is a multiple layers are covering this entire place. So this hole 4 basically, that is we are thinking that because of this graphene layer when it is coating the hole there is a damage in the graphene layer and it is just folded, and we can see the multi-layer on inside the grid if we go for a high magnification imaging. But if it is nicely distributed and it is a monolayer, you cannot see the graphene oxide layer at all in your images. So what actually it is happening, I will just go to the previous one and make a diagram, and it will help you to understand what actually happening when we are doing that. So here you have a square. What do you have? You have the uniform sizes hole, and when we are doing a normal imaging, what we are talking, this is the carbon. So if we are looking from the side of this particular hole, and there will be a carbon again, and your amorphous ice will be forming here and your sample will stay inside that. Now when you are coating a graphene oxide, so there will be a very thin graphene oxide layer, it is going to coat top to bottom. So we will just use this. So if this is a graphene oxide layer, this graphene oxide layer, it will going to cover all the holes from the top. So if you look those holes from the side, how you will see it? So here I am just showing 2 holes. So if this is your hole, right, if this is your hole, how this graphene oxide it will be distributed? Graphene oxide it will come all the way, it will go like that, it will go like that, it will go, come again, go like that and like that. So it will be always like that. Okay, if there is a second hole, the graphene layer it will go something like that, again it will go like that. So you can see a small amount depth inside and the graphene layer will be there. If you add the sample, your sample will stay attached here inside that. So there will be your sample and then it will be frozen hydrated, it will be within the ice, amorphous ice, but amorphous ice thickness it will be less because of this graphene oxide. But there is a graphene oxide layer it will be there that will give you some kind of stability. So when electron beam will pass through this sample, the sample movement it will be less. That is basically the main principle of this using the graphene or graphene oxide coated grid. So if you look at in this particular image, what you can see in this particular image, you can see that this is a graphene oxide coated grid, and if we measure the width of that graphene oxide, what I try to say that okay, so here you have a hole, here you have another hole. When the graphene oxide is going like this. So if you measure the height of this graphene oxide, you can find out based on this hole width that is basically going like that. So, there will be a high peak, then it will go all the way down, again there is a high peak, again there is a high peak, all the way it will go down. So, if you look at this particular region of that, if you measure this one, you can see it, this is the carbon, and then graphene oxide is going all the way down, and then again a peak is coming and going like that. So, if you go all the holes, it should be like that. Okay. So, here you can see this is the one hole, there is a depth, and then this is the peak area on the carbon, again there is a hole, and again it is going up. So, this is how graphene oxide is going. So, it is covering one by one hole, and there are some uniform depth inside that hole where your sample can stay inside this, and we can image that particular area. In this particular case, we measure how many empty holes are there, how many holes are coated with graphene oxide or partially coated with the graphene oxide. We can see almost 90% holes are coated with graphene oxide. Those are all coated with graphene oxide for the imaging. There will be some holes like this, those are partially coated with graphene oxide, or this may be a problematic. So, those are partially coated with graphene oxide. Those kind of holes will just do not consider. So, if we just go little bit more details to see the AFM image, how this graphene oxide layer it is looking. So, if we just consider this one, you can see this is a graphene layer. In this particular case, this is the graphene layer is coating those holes, and this is again a AFM image where you can see those are the empty holes, and these are the holes where it is coated. These are the holes which is coated with graphene layer, and these are basically the empty holes. Okay, what is the shape of those empty holes? If we empty holes, or there are graphene oxide coated grid. If you look at this particular grid, you can see that one, that okay, this is a empty hole, and you can see the overall depth here. There is some kind of depth is here. Now, we are coating this particular grid. If you look the same grid from a different angle, and this is the curve where you can see that this is the carbon area. So, this is going like that, and then there is a whole area. So, this is the whole area, again it is going straight. So, this is the depth it is showing, what is the depth of this particular hole or this particular hole or this particular hole. Now, if you look at, if you just try to isolate graphene oxide using the normal water, or if we do not sonicate it properly and try to coat our grid. So, most of the time you can see this kind of multi layers of the graphene oxide on top of that grid. So, that is basically, you can easily see that what kind of depth you are getting inside that hole, and it is not uniformly distributed. That is basically very harmful for cryoimaging, because most of the time whenever we are doing the cryoimaging, we are trying to see the details about our sample. So, we do not want any other information should come from the graphene oxide, even when we are doing the cell imaging. So, we do not want to misinterpret our data because of the multifolding of this graphene or graphene oxide. Now, if you pay attention to this particular hole, where you can see that these are the individual hole and it is coated. If you pay attention to this particular hole and this particular hole, you can see the differences here. Lots of multi layers are there, multi layers is formed here, but here most of the holes are coated with a uniform graphene oxide. Now, here you can see this AFM image where you can easily see this depth, where these holes are going something like this, and so on. So, it is going down, going up again like that, going like that, it is going. So, we are getting some kind of depth inside where we can add the sample and we can image. So this is another AFM image. You can see after graphene oxide coating how those grids should look like. Now there is I was talking about that functionalized graphene oxide grid. So functionalized graphene oxide grid, basically we are trying to use different kinds of functional group. Sometime we can add the amine group, or sometime we can add the fluorine group. So that basically attract the other samples. So one of this kind of study is that, so major difference between the graphene and graphene oxide is that graphene is oxidized with, and then there is a lot of functional group, that is OH, so epoxy group or COOH group, and that basically giving a hydrophilic nature of the grid, and that is basically help us to attract the protein molecules. But if we have a different types of functional group, so functional group is like that fluorine or amine group, so that has a affinity. So some protein may attract to those fluorine or the amine groups. So one of these study is like that. So there are the graphene oxide grade which is functionalized amine groups are there, which can attract attract nickel and Ta mediated protein, and then we can image those samples for cryo imaging. So as I mentioned that this is one of the example of the functionalized graphene oxide grid, where the protein molecule, very specific protein molecule which has affinity towards those functionalized group, they will attach with those molecule, and then we can rest of the molecule we can blot it away or wash it, and then we can prepare those grid as a cryo, and without purification we can get a very nice distribution of the of our biosamples. So these are different types of graphene oxide grid that we can use it, which can help us to avoid that air water interface problem, also the preferred orientation problem, and we can achieve very high resolution 3D reconstruction from those images. Not only that, we want to discuss little bit more about the stability of those grids, how stable those grids are. So here I am showing you one of the example where you can see this is a grid which is coated with a graphene oxide, and then we are using different kind of electron beam dose, because you guys are now familiar with the electron beam dose, right. So if you just consider that generally we are using 40 electrons total per angstrom square for 8-second exposure. Sometime we go to the 60 electron per angstrom square. So 60 electron, not electron square, 60 electron per angstrom square or 80 electron per angstrom square. Now if you just consider that 80 electron or 60 electron, and for a long exposure some 8 seconds or 10 seconds, so what will happen? So there will be a beam induced motion as well as there will be a beam damage. It is going to break the covalent bond of the protein sample, and it will break the side chains of the protein sample. So that is a problematic situation, but before that, how we want to prove it, how stable our graphene oxide grids are. So that we want to do a experiment. So here we selected different holes at a different position. So it is not we selected everything in the central, we will just selected some of this hole where you can see a very bright dot inside. That means we are exposed those holes with the electron beam and want to see what kind of damages we can see. Now here you can see this hole is exposed for a very long time, and that is why there is a bubbling effect. You can see that water molecule is started evaporating those areas. So if you see this kind of pattern, you know that the cryo-EM grid exposed for a long time and then the bubbling effect it is coming. So in this particular square we expose this hole at 5-6 different places. One place is here, one place is here, with a very strong electron dose we are exposed. This is another spot. So total this hole exposed with a 6, 7, 8 different exposure. So that is we want to see how stable this graphene oxide or graphene is. So in one particular hole you can see we are giving a exposure total 7 or 8, still entire hole is not melt. So that means most of the time in cryo-EM grid, if this is the hole, what we are trying to image, we are trying to image at the center of the hole. But if the holes are very stable, if ice will not melt and the drift problem is not there, what we can do, we can image per hole 5, 6 or 7 images, because we cannot, previously we cannot do these things, because most of the time if this is my amorphous ice and if there is a one exposure in this particular area, ice will start melting, or there will be, this area is completely exposed, that is going to destabilize this entire hole. That is one of the reason we try to avoid. But if the stability increase like that, then definitely per hole we can image 4, 5, 6 whatever it is possible, and we can cross check that it is damaging our sample or not. Here also you can see this is a hole where we just give one exposure, we give a very long exposure. Here you can see it is a long exposure, and bubbling effect you can also see that water is started boiling there basically. Okay, so this is some example where we are just a normal holy carbon grid, and this is the graphene oxide coated grid, and you can see the difference between the number of sample. So here you can see this squares, there are many protein samples are absorbed, where this is the normal holy carbon grid, quantifoil grid, very few protein samples are there. So basically graphene oxide absorbs more protein sample, and it is giving that stability. Why it is absorbing more protein sample? Because there are some functional group, and those are the charge molecule that may attract the more protein sample. One more thing, that graphene oxide grid, it has all these functional group, that COOH group, epoxy group, OH group, that make these grids very hydrophilic, so it can absorb the protein sample easily, and we do not need to do glow discharge all the time whenever we are doing the imaging. Now as I mentioned that in previous slide, that this is one hole and it is a very stable, and that is why we are giving 4 different exposure, and then we are measuring the power spectrum of that. So if there is a geo multilayer, you can see there is a missing thorn rings, and when there is a very monolayer kind of thing, you can see it is a very nicely distribution thorn ring. And we check all these different exposure here, what is the power spectrum of those 4 exposure, and all the cases we can see the power spectrum is nicely distributed. So that is basically stabilizing our images. And this is some of those graphene oxide monolayer grids, where you can see this is the Lacy carbon grid, this is the quantifoil grid, this is the enlarged view of one of the grid, this is the damage grid of that Lacy carbon coated grid. And this is basically distribution of this protein sample, how it is distributed in that graphene oxide coated grid or continuous support frame grid. And if there is a continuous support frame, there is a high chance there will be a evenly distributed protein sample, and there will be a stability. So there are total 5 points, that one, it will be the stability, and it will attract more protein sample, and then less amount protein concentration is good enough. We can collect multiple images in one hole. Third one is that most probably with the graphene oxide grid we don't have to do the glow discharge. So those are the basic plus point of this sample. So this is all about the graphene oxide grid. Whenever we are prepared this graphene oxide grid, we can use this for structural study, and we can achieve very high resolution structure, where the side views will be less, and it absorbs more biological sample. That is one of the reason very few images are required, and we can get enough number of particles. And if there is a no drift or beam induced motion, if particles are stable, less number of particles are good enough to reach a high resolution 3D structure. That is basically we are calculated, many groups are using those techniques to implement in cryo-EM. There are some kind of problems or limitation is still there. So, like getting the graphene oxide monolayer, most of the time it will be multi-layer, it is going to affect the imaging. It is not so easy target to get a graphene oxide monolayer. That is one of the major limitation of these techniques. So, I will stop here about this graphene oxide sample preparation part. So, we can use this graphene oxide sample preparation or carbon coated grid for imaging. So, this is all about the sample preparation. In next class, I will discuss about the data collection and little bit about the contrast transfer function that will help us about the 3D image reconstruction. Discuss about the 3D image reconstruction. Thank you all for your attention.